Hello, everyone, and welcome to today's webinar, Human Pluripotent Stem Cells as a Model for Neurodevelopment Toxicity. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GIBCO by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Cláudia Miranda, Post Doctoral Research Fellow. Cláudia, you may now begin your presentation.
Thank you for the kind introduction. So I'm proud to be part of the Cell Culture Hero series, and I would like to thank GIBCO and Thermo Fisher for inviting me to present my research. I would also like to thank Lauren, Chelsea, Taylor, and Michelle for all their arrangements to make this webinar a success. My name is Cláudia Miranda, and I am currently a postdoctoral researcher at the Institute for Bioengineering and Biosciences at Instituto Superior Técnico. My research is focused on developing novel human pluripotent stem cell-derived neuroprogenitors towards the assessment of potential neurodevelopmental toxicity under 3D conditions. Just a brief outline of today's session. So first of all, I would like to welcome all of you to Portugal to meet our lab and to tell you a little something about me. Then I will move on towards a scale-out approach towards the neural induction of human-induced pluripotent stem cells, either for neurodevelopmental toxicity studies or as a model of prenatal exposure to cannabinoids. Then we will move on to final remarks and some future work, and also for the acknowledgments section. So welcome to Portugal, the country where I was born, raised, and where I am currently working in. Portugal is located on the western continental side of Europe, right near Spain. Although we are a small country in terms of size, we have an immense variety of landscapes, so ranging from the snowy mountains in the center of the country to our seven-hilled capital city, Lisbon. We also have beautiful landscapes in the north, filled with mountains where we have lots of vineyards, as well as on the plains in Alentejo. We have beautiful beaches on the south in Algarve and lots of monuments to visit, including castles and palaces, and lots of history, since Portugal is one of the oldest countries in Europe. We have Porto, home of the world-renowned port wine, and we are a very proud country, especially concerning sports, and we are currently the European soccer champions. I am currently working at the Institute for Bioengineering and Biosciences, which is located at Instituto Superior Técnico, the largest and most reputed school of engineering, science, and technology in Portugal, and also a part of University of Lisbon. Técnico has 3 campi, the main one at the heart of Lisbon in Alameda, CTN, Technological and Nuclear Campus, and Tagus Park, where our lab is located. So just to tell you a little something about me and my journey, I first completed my degree in biochemistry, where I worked with human leukemia cells, and decided then to do a master's in biotechnology at Técnico, where I fell in love with stem cells. Since then, I did my Ph.D. at IBB, working on the establishment of platforms for scale-up of neural induction of human pluripotent stem cells, and I have been a postdoctoral researcher at IBB IST since 2016. So here's a quick pic of my work group at IBB, which is called the Stem Cell Engineering Research Group. And without further ado, I will move on to present my research. So my research is based on the use of human pluripotent stem cells. So these cells are very special, so they can be differentiated into virtually any type of cell within the human body, and they have the ability to self-renewal. So these cells can be derived from two main sources. We have the embryonic stem cells, which are derived from the inner cell mass of the blastocyst, and we have the induced pluripotent stem cells, or iPS cells. These cells are somatic cells that are reprogrammed back to a pluripotent stem state-like. So the main advantage of using iPS cells when comparing with embryonic stem cells is that we would not have the ethical concerns that are often associated with embryonic stem cells due to the destruction of a potential human life. Also, iPS cells give us the ability to do disease modeling in vitro, as they can derive patient-specific cells and then culture them in the lab to see how the disease progresses. We can also use them for drug efficacy testing, for drug safety, and in the future, we hope to use them for organ replacement therapy. But before we can do any of this, we still need to have large numbers of cells, and we need to obtain pure populations of the cells that we desire. And in order to do this in vitro, we have three main platforms that are used for the culture of the cells. The first one, and the most classical approach, is the monolayer, where we have cells attached to a surface, which is coated with an extracellular matrix. So this system provides a defined substrate for cellular attachment and allows a uniform application of external stimuli. However, since the surface area available may be too low, we would end up with low cell yields. And this can be surpassed by using microcarriers. So microcarriers are small particles that can be coated with ECM that provide us scalability by providing large surface areas for cells to adhere to. We may have some trouble in here because the cells may have low adhesion to the microcarriers, and there's a high cost associated, since for using the cells, we would end up with higher costs in the downstream processing because we need to separate the cells from the microcarriers in order to be able to use them. And to surpass this, we have spheroids, or 3D aggregates, which are the ones that I work with. So in here, instead of having the cells attached to a surface, the cells are actually attached to each other, providing a 3D structure. And like this, we are able to better mimic the in vivo environment, and since we are not dependent on the surface that cells need to adhere to, we may have high cell yields comparing with the other systems. And as we do not have to use microcarriers or ICM, the process ends up being more cost efficient. However, we need to take in mind that may be some size limitations, because we need to consider that if the aggregates get too big, which is around more than 300 to 400 micrometers in diameter, there may be necrotic centers in the aggregates where the gas transfer and the nutrients do not reach. So we need to take care of that. And the great thing about the cells is that in vitro, we can virtually derive the cells into any type of cell within the human body. And to do this, we modulate the signaling pathways to direct cells to the lineage that we desire. So we can end up with cardiomyocytes, with neuroprogenitors, or even insulin-producing cells. In this case, I'm going to try and force my cells to go towards the neuroprogenitor state. As I mentioned, so I use aggregates to do this process, and free-floating aggregates often have another problem, is that they tend to agglomerate if they are not under the right conditions. And in order to prevent this, we can use microells. So in these microells, we are preventing the aggregate from agglomerating. At the same time, we can also define a specific cell number that we want to have in each aggregate. And it has also been demonstrated that this spatial confinement can regulate early commitment steps of human pluripotent stem cells by accumulation of endogenous factors. So, and to derive the cells towards the neural lineage, we used a very widely used protocol, which is called the dual SMART inhibition protocol. And basically, it uses the culture medium N2B27 and two small molecules to modulate the pathways, the SB and LDN. And during the first 12 days of this differentiation protocol, we maintain the cells in the microwells. So here, you can see how the cells look like. So at day 0, we have an aggregate that's around 150 micrometers in size. And by day 9 and 12, we can see that the aggregates grew, but at the same time, instead of being homogeneous, we have some darker zones that may indicate that we have different types of cells within our aggregates. Also, by analyzing aggregates at day 12, we can see that we have expression for nestin, a neural marker, and that we have low to no expression of the OCT4 pluripotency marker, which indicates that our protocol is efficient towards the induction of neural progenitors from human pluripotent stem cells. So, and we took advantage of this platform to try and assess if we could use it towards neurodevelopmental toxicity studies. And in this case in particular, we use it to study the effect of valproic acid, or VPA. So VPA is an anti-epileptic agent used for treatment of neurological diseases. However, it has to be very careful when prescribing this medication because it may interfere in the formation of the neural tube. So during pregnancy, there may be risks of reduced cognitive functions, malformations, development delays, and autism-related behavior in the fetus. It may also lead to microcephaly, which is basically the baby being born with a smaller head size. And in order to assess this effect in vitro, we use the dual SMART inhibition protocol. We maintain the cells inside the microwell for the first 12 days. And during the initial period of the neural induction, the first 9 days, we incubated the cells with different concentrations of valproic acid. And the first result that we got is that with a range from control conditions to 1 millimolar of valproic acid, we can see that when we have 1 millimolar, almost all of the cells are dead, and that we have a decreased aggregate diameter at day 12, just a little for 0.3 millimolar, but more than half of the diameter of the control when we are looking at the 0.5 millimolar concentration. So this may be one of the first results that may comply with the microcephaly report. Also, another way that we had to assess if the cells were following the correct differentiation pathway was to take a look at the neural cell adhesion molecule, or NCAM, during neurospecification. So this marker is virtually non-existent in pluripotent stem cells, but its expression increases as we are going towards the neuroprogenitors and then increases again as we are obtaining neurons. So, and by taking a look at the graphic, we can see that in every condition we have less NCAM positive cells during neural induction, either at day 12 or at day 19, when comparing to control conditions. So we have alterations in this marker, which may indicate if we have a prevalence of neural progenitors versus neuronal differentiation. So we needed to assess this further. So we next assessed the neural rosettes, which are the in vitro counterpart of the neural tube. So we first looked at the number of neural rosettes. And what we can see in the first graph is that using 0.5 millimolar of BPA actually significantly increases the number of rosettes per million cells when comparing with control conditions, which is in line with the results that the cells are not following the correct differentiation pathway. We also assessed another parameter, which was not a number, but the radiality of the rosettes. So for this, we did the difference between the geometrical center of the rosette and the center, the physics center of the rosette. And in here, you can see in the pictures on the right that under control conditions, they have beautiful rosettes with the centers right in the center. And we have a disruption of the location of the center in the pictures as we go towards the 0.3 and 0.5 millimolar of EPA. So we have alterations even in the neurorosette number and also in its structure, which may lead in the future to more problems on the neural differentiation. We also assessed by real-time PCR analysis neural markers such as PAX6 and SOX1 and also pluripotency markers such as OX4 and NANOG. And the first thing that we are able to see here is that we have an upregulation of SOX1, especially on 0.5 millimolar of EPA. So on the other hand, if we take a look at the pictures, we can see that the exposure to VPA leads to a delay in the formation of MAP2 positive dendrite networks, where we can observe them at day 75 in the control, but we can only observe the MAP2 marker at day 125, either at 0.3 and 0.5 millimolar VPA. So by gathering these results, and especially the SOX1 expression, we can see and hypothesize that instead of the cells going from a rosette form and following the normal neuronal differentiation towards generating neurons, we have a stabilization of SOX1. And it has been already demonstrated that cells that have a SOX1 knockout have been shown to exit from a neural progenitor state faster than wild-type cells. So this high expression of SOX1 in the cells exposed to 0.5 millimolar VPA when comparing to control conditions may be arresting the cells in a neural progenitor state instead of differentiating at a normal rate. So this complies with the results that were obtained for the microcephaly because we have less neurons, so the brain will not grow as much. So this was the first platform that we used for studying the neurodevelopmental toxicity, but we also wanted to go for a very hot topic, which is the use of cannabinoids. And there has been an increased usage of cannabis and cannabis derivatives in the last few years, either for recreational purposes or for medicinal. And so long, around 113 different cannabinoids have been isolated, the so-called phytocannabinoids. So CBD, for example, is a non-psychotropic substance, and delta-9 THC is the primary psychoactive component in cannabis. So, and phytocannabinoids such as delta-9 THC or just THC can bind to the endocannabinoid system receptors, which regulate a variety of physiological processes in the human body, such as synaptic activity in the central nervous system and analgesic and metabolic effects in the peripheral nervous system. And recently, synthetic cannabinoids have been developed based on the structure of THC, such as, for example, THJ018 or THJ, and also EG018 or EG as we will call it. So the cannabinoid receptors, CB1 and CB2, are expressed in the developing brain, and there is growing evidence supporting the role of these receptors in neuroprogenitor proliferation and modulation of neuronal maturation and specification, and also the expression of these receptors in the fetal and adult brain was also reported. So prenatal exposure to cannabinoids acting as agonists of CB1 or CB2 receptors can produce long-lasting effects on the endocannabinoid system, and this signaling will affect motor activity, verbal development, nociception, and also drug-seeking behavior, among other processes. And also, due to their lipophilic nature, phytocannabinoids can easily permeate cellular membranes, passing from drug-consuming mothers' bloodstream into fetal tissues. So, and how do they work? So, CBD binds to CB1 and also has a negative allosteric modulation of CB2 receptor. On the other hand, THC binds to CB2 and acts as a partial agonist. Also, synthetic cannabinoids act as a full agonist of CB2 receptors, and they provide stronger and long-lasting effects on brain cells, which are often associated with more severe side effects and also intoxications. However, the cellular mechanisms underlying cannabinoid effects on human neural development are still poorly known. Thus, there is an urgent need for a better understanding of the impact of these substances on human brain development, especially due to the contemporary trend of increasing cannabis use. So for this research, we used a similar initial approach as we did with BPA, and the cells were cultured inside microarrays for the first 12 days. The main difference was in the period of incubation with cannabinoids, which was from day 19 to day 56. We used concentrations of 10 micromolar for every cannabinoid, including CBD, which we had to reduce to 1 micromolar since the cell did not survive. The endpoint analysis were mainly immunofluorescence, gene expression analysis, and single-cell calcium imaging. So the first result that we obtained was that by day 30, all 6 conditions showed expression of progenitor marker PAX6 and also neuron-specific MAP2 protein. Interestingly, THC, EG, and THJ treated cultures showed decreased staining for the PAX6 neural rosette marker, which suggests a possible decrease of progenitor pool and exit for differentiation. So to quantify better this effect, we performed immunostaining for HuCD protein marker that is expressed earlier in differentiating CNS neurons, and we counted the percentage of positive cells. So our data show that the decrease in the PAX6 staining is accompanied by the trend for an increased percentage of UCD cells, positive cells, upon exposure to all cannabinoids. In parallel, all CB-treated cultures exhibited elevated levels of apoptosis marker cleaved caspase-3. Together, these results indicate that both THC and the 2 synthetic cannabinoids lead to premature differentiation of rosette progenitors that seems to be more pronounced in the case of THJ, possibly due to lower neuronal survival upon exposure to THC and EG. And one of the reasons for lower neuronal survival could be a functional impairment or inability to achieve functional maturation. We next questioned if these neurons were able to achieve functional maturation. And for this, all cells were maintained in neuronal maturation medium up to day 56 with continuous exposure to cannabinoids. Immunofluorescence staining for mature synaptic protein SYN of THC and EG treated cells revealed decreased staining intensity, suggesting a lower density of mature synaptic puncta. Additionally, an increase in GFAP staining was also visible in both conditions for the synthetic cannabinoids, which indicates a premature glial differentiation in these conditions. To further evaluate both the neuron-glia ratio and the functionality of these cultures, we performed single-cell calcium imaging. Controlled and cannabinoid-treated culture cells were sequentially stimulated by exposure to potassium chloride and histamine. Differentiated functional neurons are expected to open voltage-sensitive calcium channels in response to potassium chloride, which results in a massive influx of calcium to the cytoplasm. On the other hand, immature neurons, neuroprogenitors, and glial cells express functional histamine receptors, which stimulation also increases intracellular calcium concentration. Then, the histamine-potassium chloride ratio can then be used to evaluate the proportion of mature and immature neurons in these cultures. Upon potassium chloride stimulation, there is a sharp increase in cytosolic calcium concentration observed in both controls and also in CBD-treated cultures, with an average fold change of fluorescence intensity around 2 in all three conditions. In contrast, very few cells responded to histamine in this culture, and the few responding cells exhibited fold change below 1.5, indicating that most of the cells in these cultures are excitable neurons, with just a small proportion of neural progenitors and/or glia. These results also agree with the very rare occasional GFAP staining, which was detected in both control cultures at this stage. Then moving on to CBD, exposure to CBD has a small but statistically significant effect on the functionality of differentiating neuronal cells, with a lower amplitude of response to potassium chloride stimulation when comparing with the controls. In contrast, the exposure to both THC and the two synthetic cannabinoids severely impaired the ability to differentiate neurons to respond to both potassium chloride and histamine. So delta-9-HT treated cells showed delayed response to potassium chloride, and as the majority of these responding cells either responded too late or were unable to return to baseline intracellular calcium levels, it is impossible to determine the exact proportion of cells that responded to histamine. Therefore, the higher proportion of histamine-responding cells most probably correspond to a mixture of functionally impaired neuronal cells showing abnormal response to potassium chloride stimulation, and with some glia-like GFAP-positive cells that are also more abundant in this condition. And in comparison to THC, THJ has a slightly less severe effect on the functionality of neuronal cells, where there is a less pronounced delay in the response to potassium chloride. So the percentage here is similar to the controls, but the cells are still unable to return to baseline levels after washout of potassium chloride containing medium. The percentage of histamine-responding cells in this condition is even higher than THC, reaching around 100% of all acid cells. Again, this response is most probably the result of the functionally impaired neurons with very slow kinetics, rather than of neuroprogenitors or immature neurons that express both voltage-gated calcium and histamine receptors. So finally, EG-treated cells represent the most severely affected condition, with no discernible peak of potassium chloride response and also the highest percentage of cells with weak or no response to potassium chloride. Like the THJ-treated cells, there is a continuous increase in intracellular calcium that was also observed. So this data might indicate that exposure to EG018 leads to an even greater delay in the cellular response to potassium chloride stimulation. This conclusion is further supported by the absence of a well-defined peak of histamine response despite the abundance of GFAP-positive cells in this condition. Lastly, we also did real-time PCR analysis, and we used the ethanol condition to normalize all of the results. And in here, the most pronounced fold changes of expression levels were detected for the genes encoding the cannabinoid receptors 1 and 2, which are the CN1R and the CN2R. And exposure to both CBD, THC, and EG, and at a less extent THJ, led to a significant increase in the expression levels of CN2R. Also, interestingly, the levels of the cannabinoid-1 receptor were slightly increased after exposure to THC and to the two synthetic cannabinoids by day 30 and decreased by day 56, probably reflecting the long-term downregulation of CB1 levels, which is similar to what was reported to occur at both protein and mRNA level in response to chronic exposure to THC. However, both synthetic cannabinoids, and in particular THJ, were able to elicit a stronger effect than THC. Curiously, THJ and CBD, which were found to cause less functional impairments on neuronal cells, according to the data that I showed about the single-cell calcium imaging, they led to more pronounced changes in gene expression levels in both neural markers and CB receptor genes. Thus, the effect of exposure to these two substances requires further examinations by different techniques that we use in this study, so we can note the nature of the cellular response to these substances. So in conclusion, our data show that continuous exposure to THC and also to synthetic cannabinoids can induce functional impairments to newborn neurons during the formation of the human central nervous system, which is able to produce a deep and lasting impact on the overall brain structure and function. So we will just go through a quick overview of what we talked about here today. So first, the spatial confinement using microelves is very advantageous, and it promotes accumulation of endogenous factors while avoiding aggregate agglomeration. And these microelves can be used as a scale-out approach to generate neural progenitors from human pluripotent stem cells. Also, we saw that valproic acid has a negative effect on neural development through the deregulation of normal neuronal differentiation, and also that prenatal exposure to cannabinoids, namely THC and the synthetic cannabinoids that we studied, promotes precocious neuronal and glial differentiation, as well as abnormal function of voltage-gated calcium channels. I would like to end up with some acknowledgments. So first of all, to my team at Técnico and Instituto Superior de Ensino e Biosciences and Instituto de Ensino Molecular for all the help that they provided for the studies, to my research group that is just amazing, and also for funding from FCT and Biogrammy. And lastly, but not least, to Thermo Fisher Scientific for all the amazing webinar series that are currently presenting. So thank you all for your attention, and I hope that you have some questions for me. Thank you.
Thank you, Cláudia, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. First question is, can this methodology be applied to different types of differentiation protocols from human pluripotent stem cells?
Hi, so it can. And we also have been applying this protocol using the same microwells that we use in here. We use it towards the cardiac differentiation, and we are now starting to have some data on the effect of cardiotoxic drugs, and we hope that to share the data with you soon.
Great, thank you. Okay, our next question here. Why is the exposure to valproic acid and cannabinoids at different time points?
Okay, so in these two cases, we were looking at different things. So in the first one with valproic acid, we were looking at the effect on the neural development. So we wanted to assess the usage of VPA during pregnancy, and to do so, we incubated the cells during the first steps of the differentiation, which is from day 0 to day 9. And in the case of cannabinoids, we were actually looking to see the effect of these molecules on the receptors. So we only incubated the cells from day 19 to day 56 because we wanted to make sure that at least some cells were already expressing the receptors in order to the cannabinoids to have some effect on the cells. And that's why we incubated the cells at different time points.
Okay, great, thank you. Now, is there any reason to choose that range of concentrations of valproic acid?
In fact, there is. So when we were studying the effect of valproic acid, we wanted to include a range that included the physiological concentration that occurs in the bloodstream. So we used concentrations between 0.01 and 1 millimolar, and the actual physiological concentrations in the blood are around 0.3 to 0.5 millimolar, and interestingly, those were the concentrations in which we found the negative effects of VPA on the neural development.
Okay, thank you. Next question, will you be using microwells for single cell analysis, or do you prefer seeing cells in the global context?
Okay, so in this case, we actually tested the cells using a global context because of two things. One, because it's cheaper, and that was just one of the reasons. And I think it's interesting to have, if possible, to always have a look at everything. So not all of the cells are equal, and although it's also interesting to have a look at a single cell analysis, and actually in the case of the cardiac differentiation, we are going towards that area. In this case, we only tested a global context. So, but it would be interesting to have a single cell analysis to see if the response to a small population of cells within the aggregate, or in the in vivo case within the brain, is enough to promote the same response as if all of the cells were in the same condition. So that would be interesting, and we are taking a look for that in the future.
Thank you so much, Cláudia. And do you have any final comments for our audience before we go today?
Yeah, thank you. First of all, I would like to thank again to all of you that attended this webinar today. It was a pleasure being here and showing you our research and what are we planning to do in the future in our lab. And I hope that you also apply to this program, because I think it brings a lot of attention and also a lot of interaction with people from different backgrounds. And please feel free to reach me to know a little more about our research, and so I can answer more questions. So thank you again for attending this webinar.
Thank you, Claudia. I'd also like to introduce our GIBCO Cell Culture Heroes Program Manager, Taylor Thornton, who will also provide some final comments.
Hello, everyone. I hope you enjoyed Claudia's presentation. For the latest updates on upcoming GIBCO Cell Culture Heroes webinars or to apply to become a hero, please visit our website at thermofisher.com/cellcultureheroes and follow our social media pages on Instagram and Twitter. Our handle on Instagram is GIBCO Cell Culture, and on Twitter it is GIBCO. Thank you so much for watching the webinar.
And thank you again, Claudia and Taylor, for your time today. We would also like to thank LabRoots and our sponsor, GIBCO, by Thermo Fisher Scientific for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.